It is estimated that over 90% of fungi species remain unknown to science. So how do scientists discover a new species through the process of DNA extraction? This knowledge is crucial for researchers, biologists, and anyone interested in mycology. DNA is the hereditary material in humans and almost all other organisms. Each cell in an organism has the same DNA, and it is DNA that governs the organism's functioning and reproduction. Studying the DNA of fungi is crucial for several reasons. It helps in identifying different fungal species, understanding their evolutionary relationships, and can even aid in the development of new pharmaceuticals. Fungi play essential roles in ecosystems, and the genetic study provides insights into their complex life processes. The first step in identifying fungal DNA is collecting samples. This involves careful selection of the fungal specimen and ensuring that the sample is uncontaminated and has completely dried. How exactly do you get the DNA sequence of your chosen sample? Basically, a small section of the mushroom is taken, usually from the lamella or the gills on the underside of the mushroom cap. This piece will be sealed in a small tube and goes through a long, complex chemical process that involves breaking, washing, and purifying the sample several times until the extracted substance containing DNA is separated from the lipids, protein, impurities, and the rest of the sample. After the extraction, a PCR reaction mix is added. Then commonly used techniques like PCR or polymerase chain action amplify the DNA to create enough material for analysis. While this machine is magnifying the DNA like a photocopy machine, macro and micro observations are conducted on the sample to serve as further evidence that helps discover its species. Macro observation involves analyzing physical characteristics of the mushroom on the whole, while micro observation is conducted under the microscope as features invisible to the naked eye is studied. Finally, phylogenetics is applied after the PCR is completed. You will have millions of copies of the DNA segment, which can then be used for various purposes, such as DNA sequencing, genetic testing, or cloning. To identify the species, you can upload the DNA information to online databases, such as GenBank, where nearly all other fungi DNA other scientists have discovered can be accessible. The database will compare your DNA segment to the others until you can get something akin to a fungi species family tree. This is a good way to discover if you have found a new species. It is estimated that there are more than 2 million types of fungi that have not yet been discovered. Identifying and analyzing fungal DNA is a gateway to understanding more about these incredible organisms. The knowledge gained can lead to significant advancements in science and medicine, particularly in areas like antibiotics and biodegradation. Thank you for watching.